Okay, so good evening, everyone. And it's a pleasure to introduce our next speaker, Professor David Nelson. So David is also a physicist like several of us here. He did his PhD in physics, working on applications of renormalization group in critical phenomena. I'm sure some of you understand the meaning of these words, but don't worry if you don't. So he has a broad range of interest in soft condensed matter, hard condensed matter, and also biophysics. And the relevant area of biophysics today being population genetics. And currently he is a professor at Harvard University in the Department of Physics. So David, let's get started.
Okay, very good. I'm going to share the screen here, and hopefully you will be seeing my first slide in the display mode.
Yes,
is that
working?
good, Kavita?
Yeah.
Okay, so I'd like to thank Kavita, Jain, and all the organizers for doing this school. It's an honor for me. I have visited India, and in particular, I've been to Bangalore and Mumbai and New Delhi, but it's been many years, and I'm very sorry I can't come back and visit you all in person. It's a cold winter morning here in Boston, and I wish I could be in the south of India and enjoying hopefully a little more sunshine. So what I'd like to tell you about today you can think of it perhaps as the physics of life, and it's some problems that have fascinated me for the last 12 years or so, myself and my collaborators in spatial population genetics. And this is an attempt to understand ideas that some of them go back to Darwin, but in a spatial context, and often inspired by experiments in Petri dishes. So here is a Petri dish experiment that I'll tell you about a little bit later. And there will be 3 lectures, as you probably know. I basically have about 1 hour and 20 minutes of stuff, but I expect the questions to come, and in some sense, these 3 lectures will only be as good as your questions. I think of PowerPoint as an opportunity to present slides with nice colors, but it's also, in my opinion, a kind of form of asymmetric warfare, because I can easily present far too much information per unit time. And so I will welcome your questions along the way. I may even pause and ask for questions, and don't hesitate to slow me down. If there's something that is not clear, it is almost certainly my fault, not your fault. So please do ask questions. So there are going to be 3 lectures. The lecture today will be a kind of overall introduction to spatial population genetics. At the end of the lecture, hopefully you'll understand what's involved with this little figure over here to the right. You'll see that the wave of expansion here is something that we like to call a pulled genetic wave coming out. I'll talk about Fisher waves, pulled genetic waves in a minute in more detail. The second lecture tomorrow is a study of what happens spatially associated with this phase diagram, where we have 2 competing organisms, just like we have red organisms and green organisms up here on the top right. When they can compete, they can try to interact in various ways that are more sophisticated than merely survival of the fittest. They can sometimes cooperate. That's mutualism here in the upper right-hand corner. They can also be antagonistic and secrete poisons. So that will be sort of what's going on in the second lecture. And then finally, I'll talk about a fairly recent work on microbial interactions and these range expansions. That the top is an example But what happens when the organisms are on a liquid substrate and they can generate their own flows? This is an experiment on yeast. These are all genetically labeled organisms. That's what the color tells you is a genotype. And this is Saccharomyces cerevisiae, or baker's yeast, colorfully expanding across the top of a very viscous liquid in a Petri dish. So that's where I'm going. And the original motivation for this work actually was human range expansions. Here's this little cartoon of out of Africa waves of migration, including to India, to Asia, eventually to Australia and so forth, and also into Europe. And by sequencing indigenous populations, looking at their mitochondrial DNA, typically, one can get an idea of when these various branches of the human family arrived at various parts. And of course, eventually, some of our ancestors crossed into North and South America as well. Now, one of the things that is striking about this very nice cartoon is that there are these numbers which are, typically arrival times, but there are two problems with it. One is that there are no error bars associated with these numbers. There certainly are error bars, and they're large. And secondly, it's an experiment that is not easy to repeat. You probably wouldn't want to repeat it deliberately. So it's hard to test theories if you can't tune the parameters. And so we tried to find a kind of surrogate for this range expansion, something that could be studied easily in the laboratory. Of course, not with humans, but with other organisms. And a characteristic number that's useful to keep in mind is if you have humans that are sort of wandering around and eventually establishing a Fisher wave. We'll talk about this Fisher wave a bit later. In 500 generations, the waves I'll be talking about, they go about 10,000 kilometers. On the other hand, if you look at bacteria in a Petri dish, and I'll show you many examples, those bacteria expand, and in that same number of 500 generations, they only go a centimeter. So if you don't mind a change in the scale factor of 10 to the 9th, you can explore some of these issues about what happens to spatial population genetics at the frontiers of these range expansions by going into the laboratory. And here is a view and sort of a white light, a grayscale view through a microscope of E. coli. These are these rod-shaped organisms that live in our intestines. There are more E. coli in a typical human intestine than there are people on the face of the earth. Here is approximately a monolayer of E. coli, and we're going to be looking at what happens at the frontier. That's where most of the action is happening. And I'll play this movie, and as you can see, the bacteria are dividing, and they divide with the excluded volume interactions that push them forward across this Petri dish in such a way that after 10,000 generations, they go about a centimeter, and after 500 generations, they go a centimeter, and that might take a day or two to happen. But you can look at something which is more detailed if you take these bacteria and you label them, say, two different colors. They're basically genetic labels for the plasmids that are carried by these bacteria. I'll talk much more about this later. But if you do a similar experiment now, zooming out, so there's a circular inoculation. This is the frontier, which would look approximately linear if you were close to the frontier. And as you go outwards, these two different colors, blue and yellow, segregate out, almost like oil and water demixing. But in this particular case, there's no chemical or biological antagonism between the colors. In fact, what goes on is a kind of genetic sorting, as we'll see, due to number fluctuations. And so, although it started off as a well-mixed population of blue and yellow, you get these blue and yellow sectors that happen at the frontier due to stochastic fluctuations called genetic drift. So that's, again, where I'm heading, just to set the stage. And at these frontiers, you can start asking questions about the nature of the competition at the frontier. Suppose the left side here is red and the right side is green organisms. These are like brown eyes or blue eyes, for example. They're inherited by these asexual bacteria. We won't talk much about sexual reproduction. It's just asexual reproduction of the bacteria spreading across a Petri dish. And this wiggly boundary is the boundary between red and green, and for some reason they separate. And now we can ask what happens at this boundary. And there are at least 4 interesting things that can happen. So these red and green strains could be neutral. They even though they have maybe a different fluorescent protein that's expressed and allows us to figure out who begat whom. Otherwise, they're identical. In fact, we've engineered the genomes to be as identical as we could make them. Maybe a single codon would differ from the species on the right to on the left that would just change the color of a fluorescent protein. And as we'll see, in that case, this boundary is a kind of random walk. It weaves back and forth as this range expansion goes forward in time, but just due to the fact that in one generation, perhaps the green will accidentally out-reproduce the red, and then this boundary will bend to the left. Conversely, if the red in the next generation out-reproduces the green, it would bend to the right. And so that's sort of a neutral model of what goes on in the spatial population genetics. If, on the other hand, the green can out-reproduce the red, then there would be like a random walk with drift, and this black boundary would systematically bend to the left. Conversely, would bend to the right if the red were favored. But real biological organisms sometimes are not quite this simple. In fact, one or both could secrete, as I'll show you in actual experiments, amino acids that could be useful to the other. For example, this green one could secrete an excess of leucine, and maybe the red can't make leucine so efficiently, so they will share their goods at the interface, and then the red maybe overproduces tryptophan and lets the green benefit. So that's mutualism, and that's a driver towards mixing of the populations rather than having a sharp boundary. And then finally, this point 4 here is what would happen if they were antagonists. And this, of course, also happens in the real world. There are bacteria that, secrete poisons that for which they have an antidote. In fact, there are antitoxin cassettes on the genomes of certain bacteria. So they are immune because they have the antidote, but they secrete a poison for their near relatives who don't have the antidote. And sometimes the relatives can respond by having their own antitoxin cassette. And over the course of these lectures, I'll show you what happened. So I see that Kavita has raised her hand. Would you like to ask a question, please?
Yeah, I didn't quite understand first. So I thought, I mean, these bacteria, they have, of course, you know, they divide every 20 minutes or so.
Yep.
They will die also. So how are you maintaining this boundary? I mean,
Okay.
I understand.
Thank you. Thank you. So over the time scale of this particular experiment, the bacteria never die. They stop dividing behind the front because they eventually, the colony thickens, they run out of nutrients, but they don't die. In fact, they continue to express the fluorescent proteins. They just stop dividing. So there's no death in this particular experiment, but the front just pushes forward at a rate determined by that 20-minute cell division time. So does that answer the question, Kavita?
Yeah, thank you.
Okay. Any other questions at this point? Okay. If not, I'll turn to the main subject of this particular lecture after this introduction. And what I'll tell you about in the rest of the talk is gene surfing and survival of the luckiest. And we'll be talking about mutations and the fate of rare mutants, or even 50-50 competitions between a mutant strain and a wild type strain in spreading populations. So I don't know how popular surfing is in India, but you probably know that surfing can either be successful, you can ride a wave, or unsuccessful, you can fall off. And I'd like to convince you that mutations behave in a very similar fashion on Petri dishes sometimes. This was work that happened when I started to get interested in biology about 12, 15 years ago. In fact, it was sponsored by our Center for Systems Biology. In fact, I now have a joint appointment with our Department of Molecular and Cellular Biology, which was a great help in facilitating this work, and started with an outstanding postdoc, Oskar Halacek, who now on the faculty at Berkeley, and I think he may be actually going back to a fancy professorship in Germany. The other person who was very much involved in all of this is Kirill Korolev. He was a graduate student, but he's now a professor on the faculty in physics at Boston University. So what's going on with this surfing to try to go in hopefully small steps. So the first thing to tell you about is Fisher waves, or sometimes called Fisher-Kolmogorov waves, and just talk about what happens with organisms in the absence of any explicit population genetics competition. Let's just assume that they're all the same genotype to a good approximation. And let's just go back in time to 1798, if not earlier. There's this famous model of population expansion. Sometimes it's called population explosion, due to Malthus, who I guess was an economist, but very gloomy person. And he imagined that if you had a certain population, I'll think about it as population in the test tube, in a certain volume, omega. If births outnumber deaths, let's focus on that situation. If that's all you look at, births and deaths, then it's pretty easy to convince yourself that the population obeys this simple equation where A is a reproduction rate. So that would be 1/20 minutes for bacteria, for example, in a well-mixed test tube. And this creates, as you probably know, exponential growth. That's not all that happens, though, because eventually when the population gets big enough, you run out of resources. Bad things can happen that limit the population when it gets too large. And so I don't know why it took quite so long, but 38 years later, another scientist, Verhulst, said, well, there really should be another term in this equation that reflects deaths. And so all the competition for resources or other bad things, maybe for humans like disease and war and so forth, will cause a negative contribution to the growth rate. And the simplest form you can put is just a quadratic function, but it doesn't have to be quadratic in general. And what this will do is cause the population to grow up exponentially initially and then saturate. And so, in fact, this is an actual experiment, experimental data of the biomass of yeast. So here, these bees are yeast cells growing up in a well-mixed test tube. So let's imagine it's shaken overnight, and gently shaken so that you blur out any spatial structure. And in fact, if you solve this differential equation, it can be solved exactly, or you can solve it numerically. You grow up exponentially, and then you eventually saturate at a fixed point C star, which is given by A over B. So that's the understanding going back to 1836. But I also want to add in diffusive migration, because now we could imagine that things aren't well mixed, that they are moving around in space in some way, maybe just random walking aimlessly in an environment in a search for nutrients, for example. By the way, if you try to fit human populations on the Earth, so just imagine the Earth is a well-mixed test tube, which of course hasn't been true over most of our history, but before COVID, people, you might remember, were flying around in airplanes quite a bit. And so in that sense, the human population has been mixing fairly well for the last century or so. And it turns out that around the year 2000, if you just fit human population to this curve, we reached the inflection point. And so now population growth, at least in some places, is slowing. So it can be, this model can be used for a variety of purposes, but if we want to take space into account, a simple natural thing to do is to imagine, following R.A. Fisher and Kolmogorov and collaborators back in 1937, we can define a population density, say a two-dimensional population density of bacteria on a petri dish, for example, or something else, and allow diffusion. So here's a diffusive flux associated with inhomogeneities in the population that will basically model a kind of random walk process. And so I want to now spend some time on how do we understand such a differential equation. It's a nonlinear partial differential equation. And I don't know how many in this audience have encountered it before, but for most physicists, it's rather strange that this equation supports waves. It's a parabolic differential equation, just meaning that there are two space derivatives and only one time derivative. It's the simplest, maybe one of the simplest examples of a reaction diffusion model. And it has this nonlinear term C squared. And the combination of the parabolic nature and the growth and the saturation gives rise to fascinating nonlinear wave-like phenomena. So what I'm going to do is not go through everything in detail, but I'm going to try to use this highlighter just to show you the essence of how people like Fisher and Kolmogorov attacked solving this particular equation. So Kavita, can you and the others see this highlighting that's going on now?
Yeah. Yes, I can see that yellow highlighting. Yes.
Yeah, if I were in India, and I wish I were in India and seeing you all in person, this is why getting some feedback and questions is going to be particularly important over the Zoom link here. I would be going to a blackboard. And so this is my pale approximation to going to a blackboard. Anyway, here's the equation that I'd like to walk you through and just show you some of its, I find still quite amazing properties. So, if you've encountered this equation, how would you attack it? Maybe the students would want to think about this. But one way to do it is to imagine that the population is small enough that the c² term can be neglected. So if the initial condition, let's say, is a very small value of C, say much smaller than the saturation concentration, which comes from these nonlinear terms, which is A over B, then we have a much simpler problem to solve. And at least at early times, before the population grows up too strongly, we just have to solve this linear equation. And if I have a kind of point-like inoculant, let's say we're in one dimension, and I want to solve this this equation. This linear equation, you can almost always solve linear equations, has this solution that's displayed here in this blue box. And what I've done is to take out this exponential growth that is naturally trying to occur at every point in space. And if you pull that out, what's left behind is just the solution of the diffusion equation. And you may know that diffusion by itself causes some spreading, like a square root of time spreading, and that arises on top of this exponential growth. So C of x and t spreads out diffusively, but at the same time, it grows up exponentially. And you can check that you take this exact solution of the linear problem and integrate over all of space, that indeed the overall population integrated over all of one-dimensional space grows up exponentially with this growth rate, which, as I said, in bacteria would be like 1 over 20 minutes would be a typical growth rate in bacteria. And so that's depicted down here, where in the graphically a little delta function or a little point-like injection of bacteria will spread along the x-axis at the early times, like the square root of time, but at also the height of this little bump is going to be growing upwards exponentially. So that's what happens at early times. However, that can't go on forever. As I grow up, eventually I reach this saturation concentration C star that is shown at the top. And this saturation concentration the saturation will spread. And if you just solve this equation, this full nonlinear equation numerically up here on a computer, you'll find that you reach the saturation population, and then you spread out to the right, you spread out to the left. And remarkably, although this is a parabolic differential equation, it's not the wave equation that you would use to describe sound or light. It nevertheless it behaves like a wave in the sense that it goes at a certain velocity. And in fact, one of the goals of this little detour to something like a blackboard is to determine what is this velocity, how fast does the wave spread, and at the same time, there's going to be, you might guess, an interface between where the population has saturated and virgin unexplored territory. So if this were a cross-section of a Petri dish, this is where the bacteria would be, maybe a monolayer or maybe several layers of them. And the bacteria would be pushing outwards into uncolonized areas of the Petri dish. And one of the things, as I said, that we'd like to know is what is this width in terms of the parameters associated with the actual equation. So any questions at this point before I go on to the next slide? So I see that 2 participants have raised their hand, and I'll let Kavita moderate and welcome both questions. So please ask.
Shubha, go ahead.
Sure. David, I'm
Yes.
sorry if I missed this. What exactly is Cx of t in this equation?
Thank you for the question. In this particular equation, C of x and t is a density of bacteria or a density of humans. So we might imagine if we're looking at a range expansion out of Africa, we have 100 humans per square kilometer in the populated areas. And so this is a density, an areal density, let's say, as a function of time. And so we're trying to track what is the density, not just in time, which we could do by integrating over the entire region of interest, and then we would just have the total number of humans, let's say, on the right-hand side, or bacteria, but it's an actual areal density is moving around in space. Does that answer the question?
So it's basically a density of individuals as a function of time, right?
Yes, that's right. It's a density of organisms as a function of time in either 1 or 2 dimensions.
Okay, thank you.
You're welcome. Go ahead, another question?
Yes, ma'am. Yeah, I just had a small clarification. How are we
Sure.
defining the width W here?
Okay, good. I'll get to that in detail. But in practice, you might ask, when does the population, starting here at this inflection point, when does it reach 90% of its saturation value, or when does it reach 10%? So you could say that it would be the range over which the population goes from 10% to 90%. There's some arbitrariness in exactly how you would do that, but that's one way to define the width, and you'll see that it emerges naturally. And I also see other questions, so I'll let Kavita moderate, and happy to answer.
Sachin?
Yeah, so perhaps maybe I have missed, but just by looking at the first equation, not solving it numerically, how can we get the intuition that the solution will look like a wave solution, which means that it will be
very good question,
minus
and I will answer that on the next slide. So if you'll just be patient, I'll try to answer that. But it's not obvious, at least wasn't obvious before R.A. Fisher and Kolmogorov did their seminal work in 1937.
Shubha, you still have a question? Shubha?
Sorry, I had to lower my hand.
Okay, sorry. Yeah, I think that's all for the questions for now.
Okay, very good. The most common question in Zoom, you know what the most common question in Zoom is, or the most common statement is, "You are muted." That seems to be my problem anyway. All right, so let's move on, and let's take a look and try to answer the very interesting question is, how do you get an intuition for this sort of thing, and what's going on? And so what we'll be doing is taking a kind of dynamical systems approach to this equation. And that arises if we look for solutions of the original equation on the previous slide that interpolate between the saturation. Let's suppose this is a profile of some, we hope, wave-like profile. And this wave, we'll see, it'll be moving from left to right. But well, let's just assume I have some profile. And maybe it's a hyperbolic tangent shape with a certain width. And we'll have to figure out what it is in the end. That interpolates between the saturation concentration and 0 as a function of z, but z is going to be an argument which is x minus v times t. So just like in a conventional wave equation, we'll look for solutions that take x and t for this partial differential equation, the partial differential equation on the previous slide, this one up here at the top. We'll look for solutions of the form x minus vt. And if I can find the appropriate v and a solution that satisfies these boundary conditions, where it goes to saturation on the left and goes to 0 on the right, I'll have something that moves from left to right. And then, of course, there could be something going the other direction as well, but we'll just look at one side of the expanding population. And so here are the boundary conditions that we have to satisfy. We go to the saturation concentration when this wave-like variable x minus vt, I'm calling it z, goes to minus infinity, and then it has to go to 0 on the right. So we can certainly substitute this ansatz into the equation on this previous slide. And if you do that, you'll get this equation here, which is marked with this asterisk out here. And it's a function now of a single variable, this variable z. And if you stare at this long enough, it looks like a dynamical system. Because if I regard z as a time-like variable, then, well, we can define something. Notice there are two derivatives with respect to z here. So this is like x double dot in Newton's equation. And then this here with a single dot or a single derivative might be some kind of friction. And then we can, at the same time, define a kind of momentum variable or a velocity variable that is the derivative of f with respect to z. So f is like the position of a fictitious particle. df dz is like its velocity. And so we can think of this second order ordinary differential equation. We've already made some progress. We took a partial differential equation and we've made it, we're hoping we can write it as an ordinary differential equation. It's second order in this time-like variable z, but we can rewrite it as two first order equations in terms of g and f. And so in fact, if I define this g of z in this way as df dz, then dg dz has this frictional coupling. That's this f prime term here, divided everything through by d. Amazingly, the diffusion constant plays the role of a mass in this fictitious particle. And then there's these nonlinear terms that are, of course, still present as well, that depend on f. And then I have this other equation, df dz is g. That's just my definition up here. So I've replaced this second order differential equation by two first order differential equations. And then you can sort of do a phase plot of what's going on for this dynamical system. And that lets you scan over all possible solutions, which of course have to satisfy the boundary condition. And notice I haven't specified the velocity yet. If we were just doing the wave equation for sound or light, Maxwell's equations, the velocity would fall out automatically. And here, I just assumed a velocity. We don't yet know what it is. But for any velocity, you can analyze these autonomous differential equations, meaning that the derivatives with respect to the time-like variables just depend on f and g, and look first for fixed points. And in fact, there are a fixed point at 0, 0, as you can easily check. That's this one down here. There's a fixed point as well at the saturating population, where again, G star is 0. That means the slope of this thing is vanishing out here, goes to 0, and over here on the left, it goes to 0. And then what we want amongst all possible solutions is a solution that interpolates between the fixed point value that is up here on the left. That's shown in this little phase plot here, G versus F. So there'll be a fixed point there. And I want to, as Z progresses from negative values to positive values, I want to go from this fixed point along what turns out to be a unique trajectory that that takes me along the separatrix to this fixed point at 0, 0, where there's no population and no change with Z. So the particular solution we want is just this trajectory connecting this hyperbolic point to the origin. And that's just a graphical way of seeing the solution. It doesn't yet tell us what the velocity is. You can get a different set of these trajectories for every velocity. But I now want to focus on the velocity. But maybe this is, again, a good point to pause and ask if there are any questions on this page. Okay, hearing none, I will move on.
Sorry, so
please.
Yeah, so in this this trajectory, like, are these being like one gets these trajectory after solving these coupled equation, or I mean?
That's right. So imagine you put it on a computer.
Okay.
And there are packages that'll solve these autonomous differential equations. It's a little bit like, as I say, dynamical systems, where the phase space is F, which is like a position variable, and G, which is like a velocity variable, because it's the derivative of F with respect to this time-like quantity. So just imagine you have an initial condition and you solve it. Sometimes the equations will blow up. The solution will blow up. It'll go off to infinity, and maybe it'll be attracted here. If you're on this side and you start up here, maybe you'll go shooting off to the upper right. But of course, amongst all the possible solutions to this set of differential equations, we want the ones that satisfy the boundary conditions. And that turns out to be this trajectory that I've highlighted in yellow. Does that answer the question?
Yes. Yeah. Thanks.
Any other questions? Thank you. Okay. Okay, I see other hands raised. Kavita, do you want to moderate?
Preet, go ahead.
Yes. Yes. Just a small question, like, are these like the velocities, are these very sensitive to small changes in those parameters? Like, there might be many trajectories, right? In that.
Yes, absolutely. Yeah. No, thank you, Preet. Yeah, it can be. So if we look at this trajectory, for example, if I'm a little bit to the right of A over B, I will go all the way off to the upper right. If you follow, if I'm a little bit to the left, I go this way. So there could be, depending, if you go to a fixed point, there can be Lyapunov-like exponents and so forth. But normally in dynamical systems, you can have any initial condition you want, but the ones we care about for population genetics are ones that start here and end here. So that throws out, eliminates a lot of the possible solutions. And again, the other puzzle is you can draw phase plots like this for any value of V, and the system presumably, if you solve it numerically, will select a particular V if this is going to make any sense at all. So that I'm going to talk about on the next slide. Other questions? Okay, I will go forward. This is the last of this little section of math. And again, thank you all for slowing me down. Keep those questions coming, regardless of whether I'm doing math or just trying to overwhelm you with PowerPoint. That's the solution to you know, we have a saying which is power corrupts and PowerPoint corrupts absolutely. So it's a asymmetric warfare, and your questions are the antidote to that problem. So there is indeed a velocity selection problem, which we can do this for a range of V's. You can learn something about the allowed values of V, and you can get a bound on it by looking at the right foot of the wave. And so what I mean by that is if I go up to this region here and everything is small, I can try to figure out what's going on at the foot. And there, it's a linear equation, and you can solve it with this wave-like form by just taking your ansatz and letting f of z be an exponential that goes to 0 as I go to the right. So when z gets large, it's possible there should be a minus sign here. That may be a typo, but you choose the exponential to fall off as you go to the right. And if you put this exponential solution in and just look at what happens to our original equation, which is basically over here. So if I just neglect the b here entirely, put in this exponential ansatz, but I think there should be a minus sign here. What you'll find is you notice what we put in this width. This is Greek letter xi is what I called w earlier. We allow for a width. It's something, some characteristic dimension associated with the interface. And so this xi, given a v and stuff, has to satisfy this equation. I'll let you check me on that, but if you put that in the original differential equation, this is has the form of x minus vt, everything works out, provided if I solve this for v, I now have a connection between v and this width xi. And sometimes this is called a dispersion relation. It's like a group velocity or something associated, but with what happens to be a nonlinear wave. So this dispersion relation, if I plot now, and suppose there is some width, I haven't calculated it yet, but suppose there is some width, then as a function of that width, so as I vary this xi parameter, I find that, you know, for small xi, the velocity gets very big. For small widths, for large widths, it also gets very big. And it describes this curve here, which has a minimum. And so there's a special value of xi associated with this minimum down here. And if you look at v of xi versus psi, the minimum occurs at this parameter, which is given by the square root of the diffusion constant divided by the growth rate. So that has dimensions of length. And at this minimum, we know the velocity. It turns out to be just twice the geometric mean of d times a. There is a particular velocity that is this minimum. Now, why should it be a minimum? why should this happen? If in fact, that's actually what Kolmogorov was able to show. It's actually not just Kolmogorov, it was R.A. Fisher showed this numerically. This is back in the 1930s when you had to use hand calculators to figure out things. And then at the same time, in parallel in the former Soviet Union, Kolmogorov, Petrovsky, and Piskunov published a seminal paper, and they were much more highly trained mathematicians. R.A. Fisher was a population geneticist, very brilliant man also, but they used able to show that for almost all initial conditions, you would get what we now call a Fisher-Kolmogorov, Petrovsky-Piskunov wave. And in particular, Kolmogorov and collaborators showed that the asymptotic velocity moving right or left for any initial condition, almost any initial condition, there are a few pathological ones which bacteria never seem to find. So any initial condition with finite support, which means there's a non-zero density everywhere in space or over some finite region of space, eventually locks in to one of these waves with, turns out, the minimum velocity. And if it's at the minimum velocity, you believe Kolmogorov or R.A. Fisher's numerical work, we then have the width as well. And so that width is given by this W parameter.
David, I have a question.
Yes, please.
Yeah, so first of all, so this C of xt is exponential in x minus vt. This is also an ansatz.
Correct.
I do not understand this.
Well, it's a great question because it's very subtle why this should work. But if you go back to this original equation here, and you at least work near the foot of the wave. So even though you have this non-trivial wave that's propagating, say, from left to right, at the foot of the wave, the c squared is always small.
Yeah, but that will give me the solution in blue.
Yes.
If I look at c square.
That's right.
Where I don't see x minus vt, I see x squared by t and t, right?
Absolutely, absolutely. Very good question. And this is the solution. This is the correct solution for short times for a delta function initial condition. But I now want to explore a different region of solution space where it looks like this. So it's not a delta function initial condition that I'm solving. And it's an interpolation from saturation to going to zero, and I can still take that different initial condition, not a delta function, which would lead to this result. You're quite right, and study what happens on the extreme right, where this is still small.
So that means if I start with the, so I understand that you want to start something like a step function type initial condition. Is that right?
Perhaps.
If I do that,
we could. Yeah, we could imagine on a computer starting with a step function. But I would, if I were doing it on a computer, I would recommend starting with a displaced hyperbolic tangent.
Okay, and suppose I use that and neglect c square, I should get exponential ansatz.
You should see a length. And indeed, if you do that and you neglect this term, you can put in this exponential ansatz, namely this ansatz here, probably with a minus sign in the exponential. Apologize for the typographical error. And if you put that in, you'll see that an exponential will satisfy this. It really does. And there's certainly a parameter with the dimensions of the length. If I compare this term to that term, there's a length that comes out associated with the second derivative. So you can put that in, and that will also solve it, just as this does, provided C is small. And of course, it works if I'm in the foot of the wave, but of course, this doesn't work if I change the sign and go to the extreme left side. This is blowing up exponentially, but it does tell me what's going on in the foot. So that's all that's all I wanted to say is study that foot, and then appeal to Kolmogorov. I'm not going to show it here, but you should look at his original papers, and then the conclusion is that the analysis that I've just done for the foot, which I think is accurate, can be spliced on to what goes on over here. And you get something which isn't exactly a hyperbolic tangent, but it looks like it, and it exponentially approaches 0 out here, just as in our ansatz, and exponentially approaches A over B. And that's a remarkable achievement to have shown that. And the remarkable, in my view, that in fact, bacteria can, in space, be modeled very accurately by this simple idea. Notice it would be much more complicated if we were not at this minimum, because then there would be 2 possible velocities, and then we'd have to decide which one was actually relevant physically. So having it at the minimum means there's only 1 velocity to deal with, and that's what seems to happen both in computer simulations of the original nonlinear equation, starting with a step function, starting with a hyperbolic tangent, a displaced hyperbolic tangent, and so forth. So that's the best I could do at the moment.
Okay. Yeah, I'll think about it a bit more. Thanks.
Yeah, no, it's a great question. Other questions? Can I see some raised hands, Kavita?
Amir?
Yes, so I didn't understand the quantity xi.
Okay, thank you, Amir. So the quantity xi here, it's just a parameter. So if it's part of the ansatz. So just like in if you had at some stage in your career quantum mechanics, you might have made a variational approximation to Schrodinger's equation. Here we're making a kind of an ansatz, or it could be it's an ansatz that tells me something about how things behave near the foot of the wave. And when the equation is linear, as you can check, it admits exponential solutions. And so we put in an exponential, but this xi parameter is to be determined. And
I think Amir is a biologist, so maybe I think he just wants to know what psi means. I think this is the same as the width.
Yes, thank you very much. Sorry, I misunderstood the question. Xi is the width. It's the same. I just changed notation. It's the width that we're going to be talking about here. This should be xi. It could also equally well be xi. Up to factors of order unity, it is the width. So thank you for that question. Sorry. Other questions before I go back to the slides? Okay, thank you. These questions are great. This is exactly what I was hoping would happen, and I'm very glad to have all these questions. So here's a summary of what we just talked about. Here's this equation. It does admit these kinds of solutions. And at short times, if I start with a delta function solution, let's say, rather than the one I was just talking about, which is exponential in the foot, what actually happens on a computer, and something like this seems to happen for real bacteria on a Petri dish, the population grows up exponentially due to this term here. This is before this nonlinear term becomes important. It spreads diffusively, just like in this approximate solution I had back here. So that's what I'm showing here. But now if you run it further on your computer, it spreads, it grows up, but eventually it grows up and it hits the ceiling, the saturated population. And here it is crossing over into a Kolmogorov-Fisher wave going to the right at velocity v, and a Kolmogorov-Fisher wave going to the left, velocity, say, minus v. This is a very nice, I recommend this nice book. There's actually now 2 volumes by J.D. Murray on mathematical biology, where this is described in more detail. So the takeaway message is it locks into a velocity which turns out to be that minimum velocity that I talked about earlier. That's twice the geometric mean of the diffusion constant times this growth rate. You can check that that's has dimensions of a velocity. That's good. And this is indeed the interface with that Amir was just asking about. I see some questions, some hands, please.
Yes. So the velocity which was computed from the dispersion relation, that was for this linearized equation. However,
that's
like
correct.
this third figure, like it has already, as you said, hit the ceiling. So
yeah, that's true. And so that velocity is certainly plausible that velocity would describe what's going on in the foot, because there the linearized equation works, and in this foot, what's happening is in the foot, things are growing up and they're diffusing, they're spilling over as well at the same time. And that does give you this exponential shape with a well-defined velocity. Now, why the velocity at the midpoint, at the inflection, should be the same as this is take it as Fisher did, the conclusion of a numerical analysis. But clearly, if there is to be a well-defined motion, the velocity at every point along this profile, when the top gets flat, and you can talk about a right moving wave and a left moving wave in isolation, those velocities had better be the same. If they're not the same, then there'll be wave breaking, and you won't get a self-similar solution. So if there is a simple solution of the form x minus vt, velocity has to be the same everywhere. If you assume that, you can calculate it down here, and then it will be, then you can get an idea of what happens here. That's the best I can do, other than appealing to some fancy mathematics or some numerical calculations that show that things settle down in this way. And there's also, you can check it with experiments. Thank you. Other questions? Okay, so any questions on Fisher waves? Well, I've had a lot of them, but please ask more if there are such questions. Questions on anything else? So you're a fantastic audience, because you already asked a lot of these great questions. All right, now I want to talk about something else that's important. Those bacteria that you saw at the frontier in the first movie that I played, the black and white image of growing bacteria on a Petri dish, they weren't a continuum of bacteria, they were quantized. Bacteria is not perfect quantization, but it's like the quantization of atoms and molecules in gases, for example, in the atmosphere. They're discrete entities. There may be 1 micron in diameter and 2 to 3 microns long. And I want to ask something, what about the quantization of population size? Can that affect population genetics outcomes? I haven't talked about that with these continuum equations, which treats the bacterial density as something which is smooth and continuous. There's a granularity when I get down to the smallest scales. Just like if I were doing a hydrodynamic theory of water flowing through a pipe, I could ignore the quantization of the discreteness of the water molecules up to a point, but when I get to small enough scales, the Navier-Stokes equations or some other equations would no longer apply, and we can ask whether the discreteness is important, and it is important. There's an emphatic yes here at the bottom in the context of population genetics, because after all, a single mutation is all that's needed to change the character if it's of a population, if it's a favorable mutation. So I want to now give you some intuition for quantization of population sizes. And that's shown here. This concept is called genetic drift, and it leads to some very beautiful problems in stochastic differential equations. And one way to arrive at it is to imagine a very oversimplified model of 2 different genetic species growing up in a test tube. And so let's suppose this is a cross-section of a test tube, and capital A and little a are the 2 different genetic variants. Sometimes they're called alleles. And the capital A reproduces, the little a reproduces, and they reproduce. And in this very simple model, it's called the Fisher-Wright model. But there are other models like a Moran model, which gives similar results. You can imagine that there's a fixed carrying capacity. And now they produce lots of offspring, but there's only enough nutrient in this experimental setup to have 8 organisms in the steady state. So all these organisms are produced by cell division, but most of them die. And eventually I just have 8 again. And we can imagine that process repeats. So this is not exactly how all organisms work, but it turns out sea urchins spread their gametes out in the ocean. The gametes do random walks, most of them die, and you end up with a new population of sea urchins. Here it's bacteria, and I have 8 of them, let's say, at every stage. Here I have 4 of each, but for each type, capital A and little a. And then the next generation, well, maybe it's 5 of the little a's and 3 of the capital A's. And we imagine we have discrete generations going on like this. And so how do you describe such a thing? I could have a population of 20, for example, instead of 8. This is a program which you can download and you might want to play with it. It's called Populus. And this Populus program lets you study by binomial sampling how the relative fraction of capital A and little a change from generation to generation. And so what you do is you produce an enormous number of little a's and capital A's, depending on how their cell division rates. Maybe they're a little bit different or something like that. And then you select 8 to continue on to the next generation. And you do that again and again. And it's a stochastic process, and a natural quantity to look at time t, and measured in generations, is the probability that a given allele A has a fraction f, or a frequency, fraction p, let's say, at time t. And so just like solving, if any of you have solved Langevin equations, for example, for Brownian random walkers and Brownian motion, there's a probability distribution of fraction of little a versus capital A. and it changes with the function of time. And if you just have one random mutant capital A and a sea of little a's, suppose there's 19 little a's and one capital A, and now you step this thing forward in time, two things can happen. One is if it's just one lonely capital A, it maybe goes up in its frequency initially, but eventually it becomes extinct. And if we don't allow mutations from little a to capital A, let's not allow that for simplicity, eventually things go extinct when they hit this bottom wall. Or there can be a rare event if you simulate this thing with a random number generator and binomial sampling over and over again, where it crosses this valley of death here, if it dies, if it goes out here, and then it takes over. So this is capital A taking over, which may happen sometimes. And so just like working out what happens for Brownian particles subjected to thermal noise, for example, you can write down, as Kimura did, a differential equation for this probability that allele A has a frequency, not the frequency of physics, not inverse seconds, but a fraction of the population P constitutes a fraction of the population P at time T. And it turns out it's sort of like a diffusion equation, but it's not one that, again, most physicists are comfortable with or have encountered in the past, but it's a differential equation where you differentiate with respect to P twice, differentiate with respect to time once, and then inside is a frequency-dependent diffusion constant. If this were a constant, and I literally a constant, I could bring it out and then I have a diffusion equation, which would describe the spreading of this probability distribution. But there's a P times 1 minus P inside, and then N controls the strength of the fluctuations just by ordinary binomial sampling. So you have to take my word for it. I'm not going to derive this, but in the references that go with the set of 3 talks, you can look it up. And the interesting thing is there's a P times 1 minus P, and it turns out that this is equivalent to a nonlinear stochastic differential equation. So maybe some of you have encountered random walks and Brownian motion. This is a random fluctuating thing that large numbers of particles, large can be maybe 5 or greater, it doesn't have to be infinite, 20. You can describe this just like it was some random walk with absorbing boundary conditions. Absorbing at the bottom, because when the large A variant dies out, it's gone. But when it takes over, the little a is gone. And so the noise, notice the noise goes away when P is 1 here or P is 0. And then it turns out, again, I have to ask you to accept this, that the strength of the noise, the analog of temperature, if you like, for these genetic processes which play out from generation to generation over time is 1 over the total population size. And there's other models that you can write down, like the Moran model, which it turns out you always get this kind of equation. And if you take this stochastic differential equation and go through the same process that would produce a Fokker-Planck equation for a pollen grain moving around in water, you'll get this differential equation on the right. So these are equivalent representations. And they allow us to figure out what the discreteness is doing. And the conclusion from this kind of analysis, there are 3 things to take-home messages. One is that if the populations are finite, they go to fixation for long times. If I take the limit n goes to infinity, if I take an infinite population, nothing happens because the statistics are arbitrarily sharp. And this would just be I started with 30% of fraction of population A, it would stay 30% forever and ever. But at finite populations, there's this noise, just the same noise that I have here, that drives you towards one's boundary or the other with some kind of splitting probability. It either goes to the top one or the bottom one. That's an interesting question in itself. Finite populations go to fixation. You get a particular genotype for long times because of the discreteness. And the probability of fixation, you can also convince yourself of a single neutral mutation, because anyone, if I just have one capital A and 19 little a's, they're all the same statistically in this model. If they're neutral, they don't out-reproduce each other. And so any one of them could take over. So if I have a one capital A in a sea of 19 little a's, there's a 1 in 20 chance it'll take over. And then finally, when I have these population waves, as you'll see, when I have these fronts that I've been talking about, the population is by definition small in the foot. So in the foot of this wave, these number fluctuations are inevitably going to be important. Let me show you what that means. Any questions on this stochastic part? I will wind up. So Kavita, I'm planning to wind up in about 10 minutes and then 5 minutes for any remaining questions. Does that sound okay?
Sounds good.
Okay, so what does this mean just intuitively? So here's one of these waves that we just talked about. And let's suppose there's a mutation or a just random insertion of a new genotype, a red genotype as opposed to a white one in the background. So this is the population density in that co-moving frame. So this is where I was writing C as a function of X minus VT. Here is one particular time. We're going to stay in the co-moving frame and ask what happens. And this is where we can understand what surfing means. Here's a wave, a population wave going through. Here's a mutation. And it may be that as it goes in this co-moving frame, it reproduces and it gets lucky and it takes over. That's successful surfing. As the wave proceeds, all the population will be red instead of white. On the other hand, that doesn't have to happen. Then there's a fixation probability. This thing where it all becomes red is fixation. So this is a Fisher wave with population genetics superimposed on what happens. And it could be that it's swept backwards. And now, if it gets swept backwards, it's only a very small fraction of the population instead of what it was initially at the foot. And if it isn't able to take over right away, it gets swept backwards. And now it's tiny fraction, and it probably will die out. And so that's an example of unsuccessful surfing. And so and this is how I'll end up for today, putting these ideas together of discreteness and et cetera, and Fisher waves and population genetics and competition. We wanted to find an experiment where we could study this in the lab.
David, one question.
Experiment. Question?
Yes. So in the last slide.
Yep.
So now if I have a fixation, because when we are in the tail, then the number is little n. It's not capital N, right? So where little n is rather large compared to the large population. So little n is much smaller. So therefore the chance of fixation is rather large, no?
That's right. If it occurs here, it has a large chance of fixation, even larger if it's further in the foot. Yeah.
So that is, I thought you said this does not happen often, or did I miss it?
Okay. So good question. So it depends. So it depends on where it sits in the foot. So here, if it's 1 out of 20 of the bacteria in the foot that has this red mutation, it'll take over, 5% of the time. If it's 1 out of 2, 50% of the time. And so here I'm just imagining it's already pretty small. It's super small if it occurs back here.
Right. Okay. Yeah.
Thank you. Other questions? Okay, so I'll end on this experiment. This is an experiment so simple that even I could do it with the help of card-carrying genetic engineers. And so what we did was we took 2 species of bacteria, E. coli, indicated by these ellipses, and they had almost identical genomes. This blue loop indicates the DNA, the circular DNA of E. coli. And this other loop is an extra chromosome, if you will, a small one that gives it a color, a heritable color. And so we had these 2 species, and the main loop of DNA was the same. They're identical in every respect. And the loops are almost identical, except one has a constitutively expressed, that means constantly expressed, yellow fluorescent protein, or of some color. Let's say it's green. And then this one has a cyan color. That's the only difference. They express a protein that lets us tell the 2 bacteria apart under fluorescent excitation. We can see who begat whom. And this is where I will start to close this particular talk. And so in fact, if you take a little very tiny droplet of 50-50 mixture of these 2 genotypes, pipette them down onto an agar plate. The water carrier fluid, the nutrient fluid that it came from a broth, evaporates away, and then I'm left with these rod-shaped E. coli, and then I wait 4 days, and then this is 1/4 of the colony that's grown up. It was inoculated here, and in white light, it looks like this, but it's a lot more interesting if you take the same colony and look at it under fluorescent excitation. That's what it looks like. In fact, let me give you a more global view of what happens. The colony was pipetted down here, and you see, and we chose, it was Christmas time in the US, and so we chose, I was happy to choose red and green, which are Christmas colors for these different bacteria. And so inside this white circle is where we had this inoculant. And so that was we call that sometimes the homeland. And coming out of this homeland is a circular, a radial Fisher wave. But at the frontier of that Fisher wave, the red and the green, which are neutral in this case, have their number fluctuations because the population is small and they're fighting it out. And you can also do a linear inoculation. So one night I was in the lab, and I basically sort of became Sharad Ramanathan, graduate student. He's a colleague of mine here in molecular and cellular biology, and he accepted me as his graduate student. So I was his slave for 4 months. And so one night I was in the lab, and I laid down a razor blade inoculation. I just touched the Petri dish along a line and waited 4 days, looked under fluorescent excitation, and we had this top view. And so what's going on, if I look at a side view, is exactly one of these Fisher waves with discrete populations, mixed red and green in this case, and occasionally red will take over, occasionally green will take over, and it happens in parallel all along this linear inoculant, which then grows outwards in time. Here's a perspective, if you were a dust mote sitting on a Petri dish looking at this tsunami, this tidal wave of advancing genotypes coming at you of bacteria, they would start off being mixed red and green, then they would segregate out. And so we have a Fisher-Kolmogorov, whatever, population wave going out, but then we get these interfaces between red and green. And you can imagine that these interfaces themselves could eventually, if there were a selective advantage, let's say, of red over green, become Fisher genetic waves, where red would take over from green, as well as having red and green separately advance over the virgin territory of the Petri dish. So that's what we think is happening. We don't have to have a 50-50 concentration. Here is a inoculation that I did with 95% green and 5% red. You can see that reflected in the homeland. And then there's this event that happens here on the edge where a kind of red ancestor, like the legend of Adam and Eve, there's an ancestor, a bacterial Eve, that is one of these surfing events that happens. They're rare because they have only 5% population of red. Here it's 2%. Same thing happens. Looks a little bit like lava flowing down the walls of a volcano. If I blow this up, you can almost trace back the lineage to where the founder population took over. Notice this fractal boundary between the red and the green as you come out. And if you were to just look at this, this is where I will close for today. If I just look at this event, you might say, those bacteria, they invented some transformational mutation. And that's what allowed them to expand. But in fact, that's not the case. We analyzed what happened here at the frontier, and there are no mutations as far as we can tell. And what we have is just by random chance. There's a random walk boundary from between red and green, which went to the right. Another one which just happened to go through the left. Once it expands enough, it can take over. So I think that's a good place to stop for today. I don't have to go anywhere. It's only 9:30 here in Boston in the morning, but you may be very tired, and I will certainly understand if Kavita decides we should stop here. But if there are any additional questions, I'd be very happy to take them.
Yeah, so let's just ask, are there any questions? Okay, so I think I don't see any questions for now, but I'm sure, okay. Well, there are. Okay, go ahead. Amir?
Yes, please.
Sorry, my hand was up. Sorry, I'll just do it.
Okay, who else raised the hand then?
Yes, ma'am, I wanted to ask.
Yeah, go ahead, Preet.
Yes, actually, this word, like you've written this, it's capturing the survival of the luckiest. Like, so basically, like, is luckiest related to these concepts of migration and dispersal? Like, are they same, like these two concepts, which?
Yeah, they're certainly related. So we have a migrating population out here at the frontier for sure, and that's by the mechanism of these Fisher waves that I'm talking about. But at the same time, we have a dispersal going on at the interface between red and green. And if these were a mixture of helium-3 and helium-4 atoms, just isotopes in a gas, the helium-3 and helium-4 chemically are almost identical. They would mix in the absence of a gravitational field or something. They would just mix. And here we have a combination of migration and dispersal. And one of the amazing features of these spatial population genetics problems is that they stay mixed. And next time I'll show you a magnified experiment where we have two colliding colonies. And because they're reproducing, the combination of migration and mixing or dispersal produces these somewhat surprising results. And we'll talk more about it next time, but it's basically survival of the luckiest. In fact, I can't resist showing you these pictures here. You can replay the tape of life, basically. You can replay it by just doing different inoculations over and over again. You can repeat your experiment. You can get error bars for theories. And all that I changed here was the density of the founder population. You see the speckles in this homeland are very fine-grained compared to the speckles in this homeland. And this is somewhat intermediate. And then statistically, you get this combination of what you asked about, migration and dispersal, and it produces these sectors. And so it's quite a nice and to, at least for physicists like myself, surprising outcome. I hope that's helpful.
Okay, I don't see any other raised hands. So, so I think we can wind up for today.
Very good.
Yeah. Okay. So we'll see you tomorrow.
Okay, I'll check in a little bit before as before as today, and I look forward to seeing you all tomorrow. And I thank you all for coming, and I thank you all for your excellent questions.
Thank you, David.
Bye-bye.